Hello, good evening. Am I audible?
Yes, ma'am. Good evening, ma'am.
Good evening. Before I start, could you please let me know what is your background and what is the intention that you joined this workshop?
Yes, ma'am. Ma'am, I'm actually a MS integrated student doing my biotech from VIT University,
Mm-hmm.
Vellore.
Mm-hmm.
And NGS is something new, and it's picking up now slowly. And
Mm-hmm.
I'm learning something new.
Mm-hmm.
So as a part of my curriculum, so NGS is like a new subject or a new topic which my professors are kind of teaching. So
Mm-hmm.
it's that's the emotion behind my me joining this workshop.
Okay. I'll try to give you the entire, background of it, what it is and how it can be done. So
Yes,
basically,
ma'am.
we will be covering uh, related to plants, but you can apply anywhere, okay? But yes,
Okay.
there is some segment over here which is specific to plants, but there is some segment which is common to any organism for whole genome.
Okay.
Okay. So
Okay.
so it's a 3 days workshop, and today we will look at some theoretical aspect of it, and then next 2 days we will be looking at hands-on things, practical things, so that you will be able to analyze the genome as well. Okay.
Sure, ma'am.
All right.
Yes, ma'am.
And feel free to interact at any time you want to discuss something else related to this, and you have any question, any doubt, you can raise your hand. You can put your questions in the chat box, and in between also I will give you some time that, okay, if you have any question, you can ask.
Sure, ma'am. Thank you.
All right. Uh, so this workshop is basically for the plant genome analysis. And we know that any genome, whether it's a plant or any species or any organism, basically we can categorize into two broad categories, prokaryotes and eukaryotes. So prokaryotes has a smaller genome as compared to the eukaryotic genome. And when we compare between the eukaryotic groups, then we have different segments like we have yeast, we have fungus, we have plants, we have animals, we have including human beings. So here we will be discussing mostly with the plant genome related things, because in the plants we have more elements than the any eukaryotic species. So whether it is related to metabolism, transcription, cell growth, cell rescue defense, communication, protein destination, like where the protein forms and then act at some other location in the cell, intracellular transport, how the transport happens, cellular biogenesis, energy production, protein synthesis, and ionic homeostasis. So when we analyze the genome, we all cover all these points, that how it is. And still there are many plants which are not mapped or sequenced, but there are many plants which are being sequenced till now. And in the plants specifically, there is a wide range of genome size. So it covers from the MB to GB. So we have smaller genomes and we have the bigger genome, the largest genome. So that we will try to cover different aspects in this workshop. So as we know that the plant usually contains three type of genomes. One is the nuclear genome, then mitochondrial genome, and the chloroplast genome. So all these three type of genomes, they have their own identity, their own functions, and their a set of proteins. So when we are saying the plant genome analysis, it can be either the nuclear genome, the chloroplast genome, and the mitochondrial genome, or all together three. So the genetic information which is present in the chromosome and the size of the genome is species dependent. So like human, we all across all the human beings, we have 3.2 GB of the genome size. But in plants, it is not like that. The every species has a different genome size. The difference in the size of the genome is mainly due to the different number of identical sequences of various size which are arranged in sequences, because every plant will have different type of functions, different type of identity, there are different type of genes, so that's why the size also varies. The gene for ribosomal RNA occur as repetitive sequences and together with the genes for some transfer RNA in several thousands of copies, because in plants also the repetitive sequences are more, so which makes the genome bigger, and the structural genes which are present in only few copies and sometimes just single copy. The structural genes encoding for structurally and functionally related proteins, often from a gene family. The DNA in the genome is replicated during the interphase of the mitosis. So what happens in the plants, because whatever the repetitive elements are there, they are present in multiple copies, and these copies are also sometimes present in several thousands number. And while the gene encoding, the genes which encode for the proteins, they are present in only few copies. So that's why this variation also varies between different species of the plants. So if we consider the genome size of different plants, so the Arabidopsis is the smallest plant species among all plants. So which is just the 145 MB. And if you will see, look at the Tulipa species, tulip, so it is of 24,000 MB. So this genome size you can see from 1.145 MB to 24K MB. So there is a vast difference between this. So why this is so? Because the variation in the copy number of the genes. So and the repetitive elements. So that all can vary in the repetitive elements. Basically, in the plants, we have different elements. So which we will be looking at what different type of elements are there in the genome. So irrespective of that, there are some unique features of the plant genomes. So there are huge genomes reaching tens of billions of the base pairs, and in many plants they are the polyploid. So usually we know that that genome can be haploid, diploid, or polyploid. So in most of the plant species, it is polyploidy, and it the abundance of the non-coding DNA, which seriously uh, hinders sequencing, gene mapping, and design of the gene. So what is that like? In the plants, the coding genes are less and the non-coding DNA is more, and which hinders the sequencing, like which affects the sequencing and mapping of the genes and design of the genes. So this is why the mapping of the whole genome of plant is bit difficult. Even the analysis is bit difficult because it has more repetitively elements, which is non-coding, and poor morphological elements, genetics, and the physical mapping of the chromosomes. Because it is the polyploidy, so it is very difficult to map the chromosomes. Large number of small chromosome in which the chromosome length does not exceed 3 micrometer. So usually the nuclear genome, when we say the nuclear genome, they are well organized in the chromosomes in plants. The number of chromosomes and DNA content in many species is still unknown because they are polyploid. So it is very difficult to assemble them, make in so contigs and then the assembly and so on. So it is very difficult to assemble these DNA contigs from the smaller fragments. So if we look at the ploidy and the chromosome number, So if we see the example of corn, though the genome is diploid, but we have the 20 chromosomes. While in wheat, it is a hexaploid, so it's a 6x, and the chromosome number is 42. So it is very difficult to assemble these 42 chromosomes and the 6x. So this makes this DNA, this genome is very complex and more and sequence it and then the analyze it. So plants is very complex when you do the analysis for the nuclear genome. Even not only the nuclear genome, the chloroplast is also because maximum. So we know that the chloroplast takes parts in the photosynthesis, which is the main function of any plant or the green plants. And the analysis of chloroplast itself is very tricky because it has very more variation in its genome, though the genome is very small. So how to analyze it, how to look at the variation, and how these functions vary from species to species. So that is the main objective of the plant genome analysis. And there are multiple type of analysis which can be done for the plants. So how these the genome is organized in the plants. So as I mentioned to you, there are two type of uh, elements, coding and the non-coding. So coding part codes for protein, and this portion of the genome for most of the transcribed genes is there. And when we look at the non-coding region, it has the introns, the regulatory elements of the gene. So basically these are the non-coding RNA, and these elements regulates the gene expression of the coding part of the genome. And these non-coding part has the multiple copies of the genes, including the pseudogenes. Intergenic sequences, so between two genes, that is the intergenic sequences. And not only that, but within the genes also we know that some introns are there, which is the non-coding part. So here when we say non-coding part, basically there are two type of non-coding parts are there within the gene and between the genes, and the interspersed repeats. So these repeats also mostly they are present between two genes, but this could be also present within the introns as well. And apart from that, there are many other elements which we will be looking at. What are those elements can be analyzed in the plant genome? Because when you want to identify a plant species or characterize them, it is important to know those elements and characterize them as well. Like in case of bacteria, usually 16S RNA is used for the species identification. But when we take the example of plants, there are different elements on which the plant species can be identified. The most of the plants contain quantities of DNA that greatly exceeds their needs for the coding and regulatory function. A very small percentage of the genome which encodes for gene, they are involved in the protein production. And if based on the kinetics of the genome, we can again categorize the DNA into low copy number, medium copy number, and the high copy number. So when we say the low copy number DNA, these are the DNA sequences which encodes for most of the transcribed genes, that is for the protein, and which is the functional part of the genome. When we say the medium copy number DNA, the DNA sequences which encodes for the ribosomal RNA, so which are tandemly repeat expressed DNA. And basically the ribosomal RNA, they helps in the translation, right? Transcription and translation. The high copy number DNA, this is composed of highly repetitive sequences which are present mostly in the intergenic regions. So these repeats also can be different types like dinucleotide repeats, trinucleotide repeats, tetranucleotide repeats, and this copy number variation is also very high in plant genome. And if we take the example of the gene classification, when we would like to classify a gene, so as I mentioned to you, coding and the non-coding part. So when we say gene, it is basically a coding part, so which codes for protein. So if when you look at over here, we can see that So this is the coding gene, and this is the non-coding gene. And between them, this is the intergenic region. So these are the 5' UTR, 3' UTR, and these coding genes will code for the mRNA, that is the messenger RNA or the transcriptome. So this part basically builds the transcriptome. Then it translates into proteins, and these proteins could be of different types, which can be as structural proteins and the enzymes, that is the function to perform certain functions. And when we take the look at the non-coding genes, basically these are the structural RNA, that is transfer RNA, ribosomal RNA, and other type of RNA like microRNA, siRNA, piRNA, and non-long coding RNA. So there are different type of RNAs are also there. Basically, if you categorize them, so this is the uh, small RNA, and these are the large RNA. So because this is the transcripts. So these all small RNAs can be analyzed as a part of the plant genome, because we know that there are only 20 amino acids, but in plants there are some more amino acids which can be reflect by the transfer RNA, and these are the sites of the transcription, and these are the regulators of these coding genes. So when we would like to see how these genes are getting expressed, then we can also study the regulatory genes. So if we look at the protein coding genes, there are different segments of DNA which can be transcribed and translated to amino acid. So this is the structure of any gene actually, basically, not only in the plants, but anywhere uh, in the eukaryotic genome. So basically, when we look at the gene, we have the 5' end, which is known as UTR. So usually 5' and 3' ends won't translate. These are the untranslated regions, known as 5' UTR and 3' UTR. So in the 5' end of the gene, these are the upstream regulatory region, and at the 3' end of the gene, this is the termination site of the transcription. So this is the structure, whether it codes from the plus strand or minus strand, but this is the upstream of the regulatory region, and this is the end of the transcription. So it means that the transcription terminates over here. Then from the site of the regulatory region, we have the promoter regions, and in the promoter region, we have two box, data box and the cat box. So these are the two set sequences for eukaryote from where the transcription site starts. So these are the promoter regions, and from there it is the transcription start sites, which corresponds to the 5' untranslated region of the messenger RNA. And AUG, this is the initiation codon. So this is the universal initiation codon, and it codes for methionine. Then we have the coding sequences. So this is the transcribed region. So you need to know that now this is the gene initiation and termination, and in between we have the exons and introns. So what are the coding sequences? So this total is transcribed, but the entire transcribed region will not code for the protein. Within that also there will be some coding sequences which only code for the proteins. So there are three termination codons: UGA, UAA, or UAG. So these three codons, these won't code for any amino acid. And uh, because this is the messenger RNA, so after termination, we have the signal for the site of 3' and trimming and poly A tail addition. So you can see that AAU, and then we have the poly A tail. So this is the site for addition of poly A tail. So this is the structure of any eukaryotic gene. So when we look at the protein coding gene in the plants, so plant contains about 10,000 to 30,000 structural genes. So these are the structural genes. But if we look at the overall genes, so there are 40 to 50K genes as a total. But these are the structural genes. They are present only in a few copies, and sometimes just one copy, that is in the single copy, and they often form a gene family. So they we know that there are different gene families which perform a similar kind of functions. So the transcription of most structural gene is subject to very complex and specific regulation. The genes for enzyme of metabolism or protein biosynthesis, which proceed in all cells, are transcribed more often. So in plants, it is a little different because every cell requires a similar kind of function or the regulation. So this is cell dependent, not just the organ dependent. So the genes, the most of the genes which are switched off and are activated only in certain organs, and then often only in the certain cells, they are activated might be in the xylem or phloem or the leaf cells, and so on. And many genes are only switched on at a specific time. So that is true in case even in any of the eukaryotic species, because all the genes will not be transcribed at once. As and when they require, then only they are transcribed and translate to the protein to perform certain set of functions. And at the same time, some genes which are expressed only in specific cells, not all the cells, because specific cells will have a specific set of functions to be performed. And in each cell in the plant, there will be some housekeeping gene. And these are the genes which every cell needs for some basic functions, which are independent of its specialization, because they are the plant cells, right? So it means that there are so as I mentioned to you, there are some specific genes which are specific to the cell type. There are some genes which are specific to the organs, or there are some specific genes which are time dependent. But irrespective of that, there are some genes which are expressed in all the cells, irrespective of its specialized functions. So the genes which are expressed at all the times in all the cells, they are called as housekeeping genes. If we look at the plant genome size, so when we analyze the plants, because in plants we have viruses, we have bacteria present in it. So there are different kind of analysis when we analyze the plant genome. So if we broadly categorize, so we can categorize host pathogen interaction, and the host genome analysis, or pathogen related analysis. So in the plants, we have to deal with RNA viruses, DNA viruses, the bacteria which are specific to some plant species, because the one type of analysis could be uh, how the diseases are occurring in the plants, and what different type of resistance mechanism is there, or whether they are sensitive to some components. So this kind of analysis also is performed when we deal with the plant genomes. Apart from these pathogens, we also deal with the mitochondrial genome of the plants, the chloroplast, and these are, you can see, these are the number of nucleotides. So when we look at the viruses, they are just in the kbs. Uh, and when we deal with the DNA virus, again, all the viruses are in the kbs, like just 1 kb or 10 kb. Bacteria is in the MB. Mitochondrial genome is also very small. Chloroplast genome is also very small. But what is the specialty of these genomes? They are highly variable. They are not stable genomes. And the another type, then we categorize the plant into different systems: algae, fronds, gymnosperms, and the angiosperm. So algae has the smallest genome, while angiosperms have the um, much larger genome as compared to these genomes. So when we do the plant genome analysis, we have to deal with a, different variety of the size of the genome. So that also one should keep it in mind when we do the analysis. Because of the genome size, because of the analysis, a lot more tools and softwares are required for different type of analysis. So, like say, suppose if you want to analyze the algae genome, because this is a little smaller than the angiosperm and gymnosperms, so maybe the same tools might not work when you analyze the angiosperms and the gymnosperms. So those are different, and also depends on how many chromosome or what is the ploidy of the plant species. So the tools will also differ a lot. So when we talk about the structural genomics of the plant, under this category, mapping and sequencing of the genome, expression sequencing tags, so which sequence will expressed and all, so that analysis can be done. The entire genome sequencing can be done, how these genomes are organized, and we can compare the genome between two species of the plant. That comes under the comparative genomics. So in this comparative genomics, so we can compare on multiple aspects. We can compare the genotypes, how these genotypes differ based on their phenotypes and all. We can compare between the disease status as well. So if the same two species or two genotypes have for the same pathogen infection or different status of the disease, that also can be compared. And for their different type of functions, their different type of gene expression can be compared. Also, in advanced comparison, evolutionary analysis can also be compared. Functional genomics, basically in this, the monitoring of gene expression is considered. So how these genes are expressed and tag for the specific proteins or metabolites, what their functions is, what their locations, or in which pathway they follow. So all that comes in the functional genomics. When we talk about the whole genome sequencing data analysis, so basically for any whole genome, not only for plants, but this is the general workflow for whole genome analysis. Raw read data, when the data is generated from the machine, so we get the raw data. Because this is the whole genome data, so the data will be large, and always we have to perform the quality control, because just looking at the data, we can't say that data is good or bad, whether we can perform some analysis or not. So the quality control at each step is very, very important. Then the data processing. And once the data pre-processing means if the quality is not good. So there are certain parameters on which the data will be evaluated. So we will see when we will look at the data. So if the data quality, we need to improve on our data quality. So that part is called as pre-processing. After pre-processing, we can do two type of analysis. So if I would like to draw this. So basically we can do the two type of analysis here. So after pre-processing, if the reference is available, or if reference is absent. So if the reference genome is available, so basically the analysis is called as alignment, because in this kind of analysis, what we do, we take the reference genome and align our data to this. And in this, basically this kind of analysis, we look at each position, and by doing this comparison, we know that where is the differences in this particular data with compared to the reference genome. So that's why it is known as alignment-based analysis. So, and we know that which type of variation it is, whether it is single nucleotide change, or any part is deleted, or anything is inserted in this genome. So the process is called as variant calling. By variant calling, we know that at which position the changes are there. So there are different type of variant calling. We can look at the SNP changes, that is the single nucleotide polymorphism. We can look at the SNVs, that is the structural variations. We can also look for indels, which are the short few bases of insertions and deletions. We can also look for copy number variation. So there are different type of variants which can be looked in this type of analysis. For every type of variation, there is a different set of pipeline. It is not that you can use the same tool or package for all these type of variants. There are different type of uh, variant analysis. Genome assembly, when the reference is absent, reference is not available, we cannot do the alignment on the reference. So then from the data, we need to just assemble the reads. So that kind of analysis is known as genome assembly. So once the read data is assembled, so now because we do not know what it is, so once the data is being assembled, then we need to perform the annotation of this. So like this is the assembled data from this part to this part, what this could be, and what is the function. So we need to identify the gene. So what gene it is, what is the function of that gene. So then we can find out the ORFs, open reading frames. We can find out the CDS coding sequences. So there are different type of analysis which can be performed on the genome assembly. And so these are the basic analysis one can perform on these data. And if you would like to do a bit advanced analysis, phylogenetic analysis, evolutionary analysis can also be performed from the same data. So if we look at the gene prediction, so what all gene prediction can be done. So once we have the assembly uh, or the alignment file, what different type of gene prediction can be done. So basically this is done on the assemblies. So if we can do the intergenic regions, we can predict the promoters, 5' UTR, protein coding regions. We can look at the splice site analysis. We can look at the 3' UTRs, terminal exons, intergenic regions. So you can see there are different elements which can be analyzed from the assemblies. And for again, each type of analysis, different type of tools and softwares are there. So some softwares are free to use. Some are available in the web as a web application. So you can upload your data on those web browser and they will predict the regions for you from the assembly. There are some softwares which you have to install and perform your own analysis. And for that, you need to know the coding. So there are different type of platforms, different type of tools, softwares, databases are available, which can be used freely. But irrespective of freely, if you want to do something more advanced, then paid softwares are also available. But most of the even advanced analysis also softwares and tool packages are available. Here I would like to stress upon one thing. So there are n number of platforms available which can be utilized for your analysis, but that all depends on the resources, what resources are available at your end, how much experience you have to analyze the genomes, and whether you know the coding or not. So all those factors depends on your analysis and reflects your analysis as well. So as I mentioned to you, there are many bioinformatic analysis tool. And basically, if we look at, so if we categorize our analysis, basically, we can categorize into two categories. So basically, we categorize our analysis into two parts, DNA and RNA. So basically, this is genome, and this is your transcriptome. For each type of nucleic acid, there could be two type of analysis can be done. So one is reference based, another is the de novo. So basically this is the alignment and this is the assembly. So when you have you have to align your data to, and again, there could be two type of data, short read and the long read. In plants, long read is much more common than the short read because the genomes are large, and when we have to assemble, we need to make the assembly a good assembly, right? Without any fragmentation or any gaps. So in most of the cases, it is a hybrid assembly. Either it is a hybrid assembly or the long reads now these days. But earlier, the short read was very popular. But still, if you want to do the alignment based analysis where the reference is already known, this works well. And also, if you want to do SNP analysis or indels or any kind of variation, the short read is much more specific and sensitive than the long read, because long reads are more error prone than the short read. So again, depends on your aims and objective and which type of analysis you would like to carry out, you can choose. So there are different assemblers are there. So basically this. So Canoe is for long read, nanopore or PacBio. Miniism is also for long read. Falcon is also for long read. This is basically for the PacBio. So these are the DNA assemblers. And here, if you take the top hat, so this is for the transcriptome, RNA, ref based. And if it is de novo, this is also for RNA, Trinity. And rest are for the DNA based analysis. So you can, based on your data, you can choose any of the assembler to assemble your data. And they are all the codes are available on the GitHub. They are free to use, but most, because as I mentioned to you, the genomes are large, so most of the plant data analysis is done on the clusters, that is the HPC, using the codes. But here I don't know what is your coding experience and other things, so I will show you a platform where medium-sized genome can be analyzed by your own in an effective way, whether it is a RNA-based or DNA-based, whether it is alignment-based or the de novo-based. So anything you can analyze it. So there are plant databases. They are very, very important, and you should know about these plant databases, because when you want to annotate it or you want to blast it against, you should know these databases separately than the NCBI and Ensemble. So like plant DB. So this is the plant genome DB, which can where all the plants are available over there. Then specifically wheat is there, rice database is there, Arabidopsis is there, phytogome is there, Europine DB is there. So this is for the pine species. Tree genes are there. So these are the gene databases. This is the legume information system. So some are very, very specific. If you are dealing with the legume species, then you can utilize this. If you are dealing with rice genome, rice database is there, wheat database is there, Arabidopsis database is there, and there are all well updated databases. You should be aware of these databases from where we can fetch the genome annotation, because if you would like to do the SNP analysis, you need to have the reference genome and how to take those reference genome for the plants and how to look whether the genome is available or not. Even before you generate the data, you should know what is available particular to that species or what is not available. Then only you can design an experiment and the analysis strategy for that particular plant species. So these are the some databases where you can look at the information, what is available and what is not available. And if you look at the overall workflow of the system, all the software tools and annotation databases which are available, and what all different type of analysis can be performed on the plant genome. So first we have the sequencing. On the sequencing part, we can generate the sequencing data for DNA and RNA utilizing the NGS platform, and in NGS platform, Illumina is the most common platform. Then once we have our data sequencing data, we can call for the variants. So this is from the DNA data, and there are many pipelines for the variant calling. So GATK, usually GATK pipeline is very common in clinical genomes, but for plant genomes also you can use. GATK is a set of tools. You can individually use them, or you can install GATK, and through GATK pipeline also you can use the same tools like SAM tools, Picard tools. Individually also they are available, and within GATK also they are available. So GATK is nothing. It is a set of tools which independently are also available. Then it can be like assembly based. Then the annotation, you can perform the structural annotation, you can perform the functional annotations. For these structural annotations, you can use for the repeat identification using some database and the tools. You can predict the genes. You can uh, there are different set of genes, CDS, introns, exons, which can be utilized over here. You can predict the UTRs and then predict the entire gene structure. And then you can visualize them through NCBI as well. In the functional annotation, you can blast them using different algorithm and different databases like NCBI, ProSite, PFAM. So PFAM is for the your prediction of domains. ProSite is for protein identification, protein sequence analysis, InterProScan, or you can use for homology blast search use for the NCBI. So there are different algorithms. Using them, you can predict their functions, like gene ontology. What is gene ontology? Basically, in the gene ontology, we can map the sequences for their biological functions, for their cellular component, for their molecular functions, and in which pathway they are involved in. So all that kind of functional analysis can be done. So basically, you can do RNA, DNA sequencing, you can assemble them, you can align them, you can call for the variant, you can do the gene expression analysis, you can assemble them and predict for the structural and functional annotation. So this is what all can be done from the plant genome analysis. So in the theory, I don't want to go more into detail, as we will go along with the practical analysis. Then I will keep telling you this can be done, how it will be, what it could be, and so on. So from here, if you have any question, let me know. Otherwise, I'll proceed. So you can unmute yourself and ask any question till here.
No, ma'am, everything was clear till here.
All right. So now what is important over here? So if I take a blank slide. So what we need now? Okay. So we need to have the data. So first important thing that we need to have the data, right? So from where we will get the data. And basically when we talk about the NGS, we get raw data in the form of FASTQ files. So we will also look at some file formats on time to time basis, because some files will be we will use that format as an input format for some tools, and when we use those file formats as an input, it will generate some output, and what the file output it will generate, we will look at all those file formats when we will generate them. But this is the starting point for as a beginning of the analysis, whether it is a reference based or the de novo analysis. So what is this FASTQ format? This FASTQ format is the 4-line format. So in this, the first line starts always with the @ symbol, and this is called as sequence header. And in this, what the information you will get, a unique ID for your sample, then read ID. You will have a read ID also. You will get some machine ID. You will have a flow cell ID, barcode. So we will look at when we will look at the data, what all these things are. Okay. So all these information you will get from the sequence header. Second line is the sequence. So this is our sequence. The third line always starts with the plus symbol, and this is called as quality header. The fourth line is the quality score. For each base. So in this line, a quality score for each base will be written, and this quality score is in the form of special characters. And these special characters are known as ASCII letter. And when we pass through this data for the quality control, so that tool will convert this ASCII letter, these special characters, into a numerical score. And this represents your data quality score, the aggregate or mean or average, you can say that, and that is represented by capital Q, and this capital Q will be equal to or greater than 30. So we will see why we set this as 30, okay, in some time. So coming back here, what we need was the data. So now if you have your own data, you are working on day-to-day generation of the data, then it is fine. If you just want to learn, you want to get sense of something from where we have to start. So there are some databases from where you will get the NGS data, raw data. Means you will get the FASTQ files. So we will be looking at these databases as well. Then we need to know the databases. Why we need databases for plants? These databases are very, very important, not only for plants, for any species, but for plants, it is more important because for every element, we have a different databases. So we will be looking at some databases for reference genomes and some specific databases which are important. And we will be also looking at some other annotation databases because we need to do the annotations as well, right? Then what we need a platform where we can perform the analysis. So we will be performing our analysis on the Galaxy Europe platform. So there are different servers, like European server is there, USA server is there. Now Indian server is also there. But as of now, this will take you to European server. But we can use this as a go to European server. So this is the intermediate server. We have Australian server. For plants, specifically for rice, we have a rice galaxy as well. So if you are working with rice, you can use this server as well. But this is general server. You can use any server for your analysis. This is free to use. And then I told you that we can do different type of analysis. So we can analyze the genome, which is the nuclear genome. We can analyze for the chloroplast and the mitochondria. So for all three, maybe you can use similar kind of tools, but you need to do the different type of analysis, like a separate analysis for these things. Whatever you can do with chloroplast, similarly you can do for nuclear genome as well. But in a chloroplast, it's a simple genome. No, you do not have multiple genome like a genomic element in it. But when you are doing the nuclear genome, you have different type of elements in it. So we will be looking at that. Also, for this, say suppose for these you can do reference based analysis and you can do de novo analysis. So there are different conditions to do that, when to do reference based, when to do de novo. So we will be looking at all these type of things, when to do what, right? So we will look at all these things. So basically, because the nuclear genomes are big, so we will start looking at all these data and other things. And considering our time frame, we will look at these two analysis basically, right? So we will see how we, what maximum things we can cover in this workshop. So if you have any other question, let me know. All right, so then we will proceed further. So we will go one by one. Sorry. So we will here. Okay. So first, let us see from where we will get the data, because we need to have the data to learn how to do the analysis. So first, there is a database called ENA. ENA is the European Nucleotide Archive browser. It is from the EMBL EBI. And in this, all type of NGS data is deposited over here. So this is a database. So you can see there are two search boxes. If you would like to replicate from some analysis, or you want to take some samples from the published paper, and whenever you deposit your data over here, you will those samples will be assigned for this specific unique ID, and the project ID will be also assigned. So if you are taking any published paper, every data is published actually, but there will be some data which is not open. Maybe the researchers or the authors have not published, but they deposited their data. So you need to know which data set is publicly available, which data set is not available for public use for some time. So that also you need to know from here. So if you have any project ID or sample accession ID, directly you can enter over here and then view it. So those IDs will be like SRR, SRX, or PRJ number, something like that it will be. So directly you can put your IDs over here and view that sample data, whether it is available here or not. But if you just want to search something, so like if you want to just something search like as a plant whole genome. So again, plants is very broad. When even if you will search plant whole genome, so you can see how many runs and other things are available. So plant metagenome will be available, plant whole genome will be available. So many data sets will be available. So to narrow down your search, you can even give the species name. Say suppose you want to search for tomato. So you can give the plant name for which you would like to search the data. And now you can click on either experiment, study, or project, and then search for it. So you can see that this is the whole genome sequence of something. So if I go here, so here also something will be there. So seed-borne bacteria, right? So you can see what it is: Bacillus subtilis, genomic random. So this is the maybe a plant genome, right? So you can just have a look, and then maybe go back, go to experiment, study. So now here you can see this is the whole genome DNA methylation, mutants in the tomato. So whole genome sequencing of root growth mutant in tomato. So look at this, what it is. So the root system is essential for plant growth and development. Manipulation of root system may improve water and nutrient capture under the normal and extreme climate condition, with the aim to initiate the genetic dissection of root development in tomato. So they might have manipulated the root system for better development. So they screened a highly mutagenized microtome collection of search for mutants affected in several developmental traits during the early growth, focusing on specific root system architecture traits. So they would like to see the traits between different growth and development of the roots. So you can see that this is the Solanum lycopersicum, that is tomato. For each sample, you can see only one file. So this is the single end data. So what is the single and paired end data? So say suppose this is my fragment. If I sequence this fragment in both the direction, so this is the forward, this is reverse. So this type of sequencing is known as paired end sequencing. So if you are doing the forward and reverse sequencing, you will be able to see _1, _2, or _R1, _R2. Means you will be able to see two files for each sample. But if your fragment is sequenced only in one direction, it means that this is the single end sequencing, and you will get only one file. So if we go and look at over here, so you can see that we can see only one file per sample. So it means that this is the single end library. So even here, if you will see, this is the whole genome sequencing root growth mutant and some project ID, date when it is publicly available, and when it is updated. Here, if you see, there are a total of 8 samples. If you move further to the right, you can see this is the plant sample from Solanum. This is the plant sample. So here you cannot see what different genotypes or what different systems is there. But if you can copy this particular project ID, there is another database. You can go to NCBI. So ENA is very good for fetching the raw data, but equally NCBI SRA is also good. So these two databases, whatever the data set is available in one database, in another also it is available. So you can see over here, and here you will get a little bit more, so you can see that this is the wild type and this is the mutant. So this part was not there. So if you can click on any one sequence of any one sample, then go to all runs. So this is the way also you can go, and you can Google for sRNA selector and then search with the project ID. So there are different ways how you can go to SRA, but ultimately it will go to the SRA run selector. So there are many things which you can do, and you can look at even SRA. There will be filters, you can filter the data. So you can see this is the project ID. So right now it is showing only for one sample. Just show for the search for the project ID. Then you will be able to see all the samples together. So you can see this is the WGS data, fast QC, RN, SRA run. This is from the Europe, Spain. This is the paired and data, genomic, Solanum lycopersicum, a whole seedling. And here you can see this is the, all the samples are here, and each sample has this much of data. And here you can see every sample is tagged with the wild type and mutant. So you can compare. So this is the comparison. So we can look whether the next step would be to see whether Solanum lycopersicum reference genome is available or not, where it is available, and so on. So when you have wild type and mutant, and it is a DNA library, so what you can do, you can look at the SNP analysis, basically the variant calling, and you can see what the differences are there between wild type and mutant. So many things can be done over here, right? So when you align to the reference genome, you will be able to see the clear differences between them, and then you can compare between wild type and mutant genome. And this is what how you can look at the data. So first you can look in ENA and then come to SRA. You can search in SRA and then go back to your ENA. So both the ways it's possible how you can search for your data, right? So both ways you can go. So first you can search in ENA, or first you can search it over there. Now we have to look whether the reference genome is present or not. So again, go to first NCBI. In NCBI, whatever the genomes are available till date for any species, any organism, any category, all are here. So this time you can select genome and you can write the species name for which you want to search. So it will take you to the genome page. If you will come down, so you can see there are different type of genomes are available. So if you look at your data, where is my SRA? So you can see this is the wild type and other things. So you can see this is the cultivar microtome. And if you come back over here, so that is the microtome, right? So this is the cultivar microtome. So if I want to ask you which particular reference you would like to choose for this data set, this is the more appropriate data which you can choose. And but this is the latest assembly version which is available over here. So if you look at the entire table, you can see one green check over here, and this is the latest one which you can see. But this is more closer to your cultivar, either this or this, anything you can choose. And if still you feel that you can use this, you can take this as well. So if you click on this green button, then you can see there are, it will show you the entire data, what is belonging to that particular assembly. So this is the genome size, this is the gap length, ungapped length, number of chromosome, number of organelle, and so on. What is the, how this genome is built, what is the sample is being used, how the data is being generated, ref-seq annotations. So this is the karyogram of it. And so here we have 12 chromosome, one mitochondrial genome and one plastid genome. If you want to take this particular genome for the analysis, you can go to FTP directory. Then this page will open, and here you can see this is the genomic fna.gz. So this is the reference genome of this particular species. So this is the file, and here you can see they have the GTF file as well, genomic GTF file. This is the annotation file. They have the protein file also. They have for the RNA sequences as well, means that is the coding file. Even they have the gene ontology also. So there are many things which you can do. So they have the translated CDS reference sequences as well. Uh, and they have the GenBank RNA sequences as well in the GenBank format. So whatever you want to use, so different tools will use the different file system for annotation and other things which can be used. But this is as a, this is the genomic FNA, this is the genomic reference genome file, and this is the RNA, means transcriptome reference genome. And this is one database from where you can take the reference genome. Another is the ensemble. So in the ensemble, we have plant database separately. So if you will scroll down, you have ensemble plants. Click on it. It will take you to the ensemble plant genome database. So you can see Rhabdopsis, Oryza, Triticum, Hodium. So this is all plant species are here. So if you will come down, we will have the Lycopersicum. So you have to just search somewhere. Solanum, Lycopersicum. This is our tomato. So this is the genome page. This is the download DNA sequence, reference genome. This is the FTP directory of ensemble. Here you can see chromosome wise file as well, and DNA top level far.gz. So this is the reference genome file. And if you go here, so this is the genome annotation file in the GFF3 format. So this is the GTF file which you can take it. And these are the chromosome wise GTF file. And this is all together, all chromosome GTF file. So this is how you can take the GTF file from here, annotation and GTF file. So NCBI and Ensemble can be used to retrieve your reference genome and the annotation file. Yes, Adarsh.
So here the reference won't be specific to a particular variety.
But you have the same species, right?
Yeah, but
So
the first one,
reference genome cannot be built on the variety, right?
okay.
It is for the species. You
Huh.
can do so, like as I mentioned to you, like you have a wild type or a mutant or something. So that is your variation. Then you can look at the variety where the differences are there between two variety by comparing the genomes. So varietal reference genomes won't be there. It is a species-specific reference genome.
So the project file which you opened, it was of a particular variety, and then in
Particular species, not for variety.
okay,
You understand? We have human beings, right?
yeah, yeah,
So human is a species.
uh,
Humans, we have every individual. So you can say that these are the variety of humans, right? So we have only one genome,
yeah,
right? And every variety or every individual we compare with the reference genome. So here also the variety goes like this. So we can align each variety. So when we say variety, what is the meaning of variety? It means that they are different from uh, like two different varieties, right? And how they differ, whether they are differ in their nutrient content, whether they are if they are flowering plants, they are different in their flowers, their colors, and so on, right? That's why we say it as variety in plants, right?
yes,
So, but you will have the reference genome, one reference genome. So if you want to see what is the difference between these two varieties, you can align your both the variety genome to this reference genome and see where the differences are. So why we make different varieties? First, we identify the markers, maybe for high yield or some disease resistance mechanism, and then we make the hybrid and then call it as a different variety, right?
yes,
So that's why we are saying, why we are talking about this particular experiment. So what they want to do here, what they want to do here, because they manipulated the root system for their plant growth and development, because that is the essential system, right? So they manipulate their root system architecture so that the plant growth and development will be better. So you can consider them two cultivars. So till they don't release them as a variety, they, until unless they are not different from each other, they cannot be released as variety. Till that time, they will be known as cultivars. So when they see that, okay, yes, your wild type and your whatever the mutant or hybrid you have created, they are really different from each other, they can release them as a variety, right? Till that time, they will be called as either cultivar or genotypes or whatever it is, right? Is this clear?
yes,
Yes, ma'am.
Any other doubt in this? So usually in plants, what happens in the field, in the lab or the smaller scale field, when they do the experiment, they do the cross hybridization and generate the different generation of plants. Then they select those which are different from each other based on their phenotypes. But on the genotypes also, they need to be different, right? Whatever the they want to see the differences, whether it is on the plant height, whether they are better in the um, uh, yield, whether they are better in the nutritive values, whether they are resistance for a specific disease or whatever it is. So they grow some, uh, different genotypes in the field, and then they grow them on a larger scale. They produce pure lines, and then when they see that yes, those pure lines are really different from each other, then they release them as a variety. So these are the methods where you can see really differences between one genotype to different another genotype, and then you can call them as a different variety. Right? So we had looked at the data from where we can take these data files. These are usually a big files. So the extension will be fastq2.gz. GZ means they are in the zipped format. All the tools can accept the GZ format, and the basic extension is fastq. And they are all labeled with their sample ID. And then we had looked at two databases from where you can take the reference genomes. Now we will be looking at what the platform we are going to use. So we will be looking at Europe, Galaxy Europe. When we go here, this is the platform where we can perform our own analysis. So let me just sign out. So when you come to this particular page, I'll put this link in the chat box. So when we are here, you can do not have an account, register here. A small form will open. You can put your details over here and click on create button. When you do that, you will receive one mail from Galaxy team with the activation link. You have to click on the activation link, and then you are ready to use your account. Come back to login or register, put your mail ID and a password, and then click on login button, and then you will be here. So then you will be ready to use your account. So until unless you will not activate it, you cannot use your account. Every account comes with 250 GB of space with 8 ports. This is the GUI based server, but at the backend it is a Linux server. So here you really do not need any coding. All the tools are integrated on this server, whatever is required for your analysis. And it's really a very good server for the students, for academician, or for anyone who would like to learn genome analysis by their own. They would like to process their own project data, and a small data set and other things. Not only plant genomes, any species, any type of NGS data can be processed over here. But just you need to take care that it should be within the limit of 250 GB. Once your analysis is done, you can download the analyzed file, store it, and delete from here. And again, you can make that space free from here, and you can reuse it. So it is not that once you fill 250 GB, you are done. No, you can delete the project from here, and then you can make that space free for another project analysis. So this is a very good server for your analysis, day-to-day analysis. Until unless you are not dealing with 50, 60 or thousands or a number of samples, like 5, 10 samples, very good analysis can be done. Visualization, analysis, end-to-end analysis can be performed over here. So here all the tools are here. And here whatever the input outputs will be there, everything will be generated over here. You can clicking by on the plus symbol, you can create a new project. Here you can switch to another project. Here you can delete, you can copy, you can share the project, you can publish your protocols. There are many things which can be done. If you go to workflow, There are different systems for the workflow. So there are some public workflows available for different applications. You can use these workflow directly for your project or analysis. You can search and if somebody shared the workflow with you, you can click on here, workflow shared with me. If you have your own workflow, you can see under this. So there are many things which can be done over here. And there are some data libraries over here, which you can use them for your learning purpose and other things. That's it. And we will learn how to use it, what needs to be done, what different things we can do when we will do the analysis over here. So today I will stop here, and from tomorrow it will be completely hands-on. If you have any question, you can ask now. So in this workshop, we will take one chloroplast genome and we will process through the assembly, and the same data which I showed you for the tomato, wild type and mutant, we can take that and process as a reference based, because the reference genome is available, and there we can see how to do the variant calling and other things. So some of the stuff from both the pipeline analysis we will be able to see. So if you have any question, let me know.
No doubts, ma'am.
All right, so we'll meet tomorrow. And this video link will be uploaded, a video will be uploaded to YouTube, and the link will be shared on the WhatsApp group. Every day it will be like that. And these videos recordings will be available till 3 months. So please make your own notes. You can watch them as many as number of times, but you cannot download them. And after 3 months, you cannot access them. So whatever you want to make the notes, please make your own notes from these video recordings.
Sure, ma'am.
So we will meet tomorrow at the same time. Till then, bye.
Thank you, ma'am. Bye, ma'am.
Bye.